if that's okay with you.
Yeah, that sounds great.
Okay, very good. It's my great pleasure to have Marinka Zitnik visiting us virtually from a very unknown university north of New Haven. You know, she has done some very amazing work at the intersection of AI and biomedical data. And today, she's going to tell us about all these interesting things that she has been doing so far. So the floor is yours.
Thank you, Amin. Thank you also for this kind invitation. It's really nice to be visiting Yale, although virtually. I hope I'll have a chance to visit it again soon in person. So today I will talk about infusing structure and knowledge into biomedical AI algorithms, and I'll give an overview of some of the recent algorithms we've been developing in the lab, and also how those algorithms enabled a few, I think, quite exciting applications in biomedical sciences. So the framing for this talk relates to graph representation learning. This is emerging as a leading paradigm for deep learning on network structured or graph structured data. We have seen that algorithms based on graph representation learning have really tremendously successful in a variety of different domains, including network biology, drug discovery, which is what I will focus on today, but also many other domains, including recommendation systems, simulations in physics, knowledge graphs, and so on and so on. And so the key idea of graph representation learning algorithms is that they take as input a particular a network or a graph. That graph might be rich in the sense that it might be attributed, meaning that nodes and edges might have additional side information. It might be heterogeneous in the sense of that nodes and edges indicate relationships that carry different semantic meaning. But now those representation learning algorithms take that graph as input, and they specify often nonlinear transformation functions that can extract useful topological patterns from that graph in order to produce accurate predictions. And so the fundamental change between modern graph representation learning and more traditional work is that we can now extract those important topological features that are most useful in prediction in an automatic way, rather than manually engineer those useful features. Now, when I say graph transformation functions, the kind of transformation functions that are very popular nowadays are based on graph neural networks. And so the idea of graph neural networks is that we started with the input graph, which I'm showing here on a very small example on the left. And graph neural network is now a function f that is a nonlinear transformation function that will tell us how we can take each and every node in that graph and its some of its local neighborhood and map that node into some d-dimensional vector space or embedding for that node. And we do that for each and every node in the graph. And in the way this is done is that this particular function f is essentially an effective non-linear propagation algorithm of node features along edges of the graph. And it's doing so in such a way that if two nodes on the left, on the input, have similar local neighborhood, they would be mapped to points in the embedding space that would be close together to each other. And so what does that mean that two nodes have similar local neighborhood? That now can be quantified in a variety of different ways. And that's the reason why we have this entire field of graph neural networks with nowadays probably tens or even hundreds of different architectures that specify in different ways what is and codify similarity between nodes in the graph, and then try to capture that similarity in the embedding space that they produce. And so in my recent research, we have shown that GNNs can be quite useful across a variety of different kinds of networks we encounter in biological and medical applications. That ranges from patient networks, where we are looking at networks where nodes are patients and edges indicate different kinds of interactions between patients, like friendship or familiar relationships, to networks that capture hierarchies of cellular systems. Those would be networks where nodes are different biological processes or pathways, and edges that are hierarchical indicate how those biological processes come together to form larger functions in the cell and human body, all the way to interactions between genes, or most recently, GNNs have been very successfully used for problems that model interactions between individual cells in human body in the case of single cell networks. And with that in mind, my plan for today is to talk about two specific vignettes. So in the first part, I will talk about an algorithm that allows us to infuse structure into biomedical AI in an effort to enable applications where we have very limited amount of annotated or labeled data. So the algorithm that I will describe allows is a general purpose algorithm for few shot learning on graphs, and applications I will cover relate to how this algorithm enabled us to identify therapeutic opportunities for new pathogens. In the second part of the talk, I will focus on ways to integrate and infuse domain-specific knowledge into algorithms. And I will cover subgraph neural network architecture, and then for the application, I will mention how we can use those methods for diagnosing diseases. So with that, let's start with the first part. So the motivation for that part is really asking couple of questions on what makes biomedical data so unique. Are there any specific properties or problems and challenges that are unique or crucially important in biomedical domains that require us to develop algorithms not in a way that goes beyond just applying them from those algorithms that were developed for other domains? And so if we think about that, there are several dimensions that make biomedical data unique. I will mention two dimensions today. So the first one on this list is that very often predictors that we want to build are used by users that might be healthcare professionals or scientists who often make very important high stakes decisions based on those predictions. So that means that we need algorithms that are robust, that can behave responsibly and trustworthy. And for those algorithms, it's not enough anymore to be optimized solely for accuracy. Other criteria are important too, and in particular, the ones that we'll focus on today relate to how to produce predictions that are also explainable and interpretable, so that they can lend themselves readily to downstream experimentation in the wet lab and are much more actionable than what would be otherwise possible. In addition to these domains being high stakes domains, I would argue that many problems at the frontier of science and the frontiers of biology and medicine are problems where we have very little label data available. So that practically means that we have very few examples that are labeled that one could use to train standards, and one could use with standard supervised learning frameworks. And because of that, it's important to have algorithms that can learn with very little label data available. Why is that a challenge? That's a challenge because many of the recent breakthroughs in AI have depended on lots of label data available. That has led to tremendous progress on pre-training very large models, such as those in language or in computer vision, and then fine-tuning those models for specific downstream tasks of interest. And sometimes that is not directly possible in the context of biology and medicine when we are working with, when we are thinking about particularly new problems at frontier of science where little label data is available. So what exactly I mean by that? So the crucial challenge here is that we are often interested in in having models that can generalize to particular new phenomena. And so generalizing to new phenomena is incredibly hard for machine learning models. And examples of that include novel drugs in development. Think of when there is in the discovery process where a new drug is being designed from scratch, there is very little data available about that new drug. Obviously, that's why because it's being developed right now. However, we might have drugs, some information available about other drugs. It is a very prominent example can be are also emerging pathogen or novel or rare diseases that we have never seen before. And of course, for those diseases which we would want to understand better, there is very little label data available for them. And so the challenge with graph representation learning algorithms is that prevailing methods and prevailing graph neural network architectures assume that what we have these networks that are already richly annotated. Like an example, what I'm showing on the left, where we have a network, most of its nodes are already labeled. And the goal is to take that label data plus graph structure into account to predict labels for the remaining nodes. And that's what the setting that prevailing methods assume. However, what happens in real world, very often we have these large networks of dark matter that have nodes that we don't understand well, whether those are genes or pathogen or drugs that we don't understand well or are currently being developed. And we have very few, a handful of labeled examples to learn with, to learn with. And so to address this problem, I will talk today about few-shot learning for graphs. So just one slide of a background. What is few-shot learning? So few-shot learning is a particular instantiation or a sub-area of a field known as meta-learning, which is in its own field in the which is a part of supervised learning. So few-shot learning is concerned with a situation of developing learning models that can make predictions. In a simple case, they would be classifiers where only a handful of labeled examples called shots are available for learning. So the terminology is that when we say k-shot and class classification, that means that we are operating in a setting where we want to have a predictor that will be able to predict n classes, where for every class there are only k-labeled examples available for training. So what I'm showing here in this particular slide is a way that few-shot learning can be implemented in practice, and it is by training models in two phases. The first phase is called meta-training phase, and the second is called meta-testing phase. During the meta-training phase, in the models, and here's an example from computer vision, are primed and tasked such that they can very quickly generalize from one task to the other with very few labeled examples. So here I'm showing one training task one. The model would be asked to construct a classifier that can distinguish between 3 classes, and for every class it just has 2 labeled examples, 2 images available. And so then we have another task where it's of similar nature, 3 classes, 2 labeled examples for every class. And so the models are trained on a large number of training tasks. And each training task is designed such that it has very few labeled examples available for each class. We are doing that because we want to achieve that our model will be used to seeing these very hard generalization settings where it has limited amount of label data. This is because when in the meta testing phase, at the test time, imagine we need to build a duck versus dolphin versus chicken classifier. We would be able to do that having only 2 labeled examples, 2 images in this case available for each class. And so few-shot learning makes this possible. And so while I'm showing this example here for images, we really want to have an approach and methodology that will allow us to operationalize few-shot learnings on graphs as well. And so that's what I will describe next. So The project I will outline, it's called GMeta. It's a general purpose approach for few-shot learning on graphs. The details are described in a paper we published at the last edition of NeurIPS, graph meta-learning via local subgraphs. And so let's start by a kind of a brief outline and mapping of the graph setting to what we have just seen in the previous slide with images. So the idea is now that instead of classifying images, we will operate with, we are tasked with the prediction of labels for individual nodes. And again, it is in the meta training phase, we define a large number of training tasks. Those training tasks are all defined on one type of labels, in this case, the Y label set, the yellow label set. In each task requires the model to use only a few labeled examples to make predictions. And I will explain how that can be achieved. And so in the meta testing phase, then we can build a completely different classifier on a different label set Y star, having only 2 labeled examples now available for each of those blue label set. So the problem.
So Marinka, can I ask you a clarifying question? So here you assume that the nodes have labels, not the graph.
Yes. So for the purpose of today's talk, I will describe the methodology as it available for node classification. So nodes have labels. We're predicting labels for nodes. And this is how my problem will be formulated. In the graph, in the paper, we are distinguishing between 3 different kinds of problems that allow us to predict labels for nodes, address link prediction, as well as graph level classification.
Thank you.
Okay, so the formulation is the simplest problem formulation of graph meta learning is as follows, and that's what I will be covering in these slides, is the following. Let's assume that we have a single graph. We have a graph G, and there is some distribution of labels given for that graph. So given that graph G, the goal is to take a label set Y that is given to us, and then use that label set during the meta training phase to train a graph predictor or a meta learner that will be then available, that will then be able to generalize very quickly to a new label set, Y star, that does not overlap with any of the labels in the label set Y. So this meta learner that we will want to learn needs to classify this unsealed label set by observing other label sets in the same graph. So the setting here is constrained with to a single graph where we're solving a node classification test problem. What is the key idea that is behind GMeta? So remember, the key challenge is that we have very few labeled examples. Because of that, kind of standard GNN architectures fail. Why? All those architectures would propagate label and feature information along edges of the graphs, but we have very little label information, so we cannot do that in a reliable way. So the key idea that GMeta puts forward is that let's use local subgraphs. What I mean by that is that in addition to using label information, which is very limited, we will also rely heavily on structural information. And so what I mean by that is that for every task, we will learn a function that we'll call subgraph signature function that will act as a descriptor of every task. And that subgraph signature function will be used to effectively transfer information from one task to the other, so that when we go to a new task, we will initialize it with the signature function that we just learned on the previous task. Why would we want to use these local subgraphs that are defined around labeled nodes? So our reasoning was the following. If we want to, in addition to using the labels, using the local structure around the labeled graphs, we wanted to understand, well, is that really sufficient, or would we need to learn the structure of larger and the functions of larger parts of the entire graph? And so what we looked at is we studied the influence that a particular node might have on the target node that we are predicting, that we want to make a prediction for. And it's possible to show that the standard graph neural network architecture are such that the influence of one node on the target node we want to make prediction for decays exponentially as we go further away from the target node. The implication of that is that it is sufficient to focus on local or enclosing subgraphs around target node to define those subgraph signature functions, and they preserve near the same feature information as the entire graph. So if I go one slide back, that means that for those handful of labeled examples that we have, we will extract enclosing subgraphs. Those will be small local subgraphs. And what we will do is when we map through function f, the label of this node and to this embedding space, we will also do that by considering the structure that appears in this local enclosing graph around labeled nodes. And so that will allow us to bring in much more information to the embedding space than simply focusing on labels. And
a quick question, something I realized I missed or forgot, which is, are you assuming that the nodes have annotations or vectors associated with them before you start doing all this training?
That's a great question. No, I'm not assuming that they have vectors associated with them. If they don't have these vectors or annotations, the prevailing approach today is to initialize embeddings with random Gaussian vectors, and then use those as embeddings in zeroth layer of message passing, which is essentially to initialize embeddings for the nodes. Alternative approach is to use one-hot encoding vectors for every node. That approach performs well, often even better than random Gaussian vectors, but it does not scale to very large networks. That's why for large networks today, the strategy is to use random Gaussian vectors as initial embeddings.
Can I also ask a clarifying question? What is the influence of a node to another node?
Yes, so that's a great question. So the influence of a node on another node is defined by the contribution of the embedding of, so if we have two nodes, U and V, so the influence of V on node U is the contribution of embedding of node V towards embedding of node U. And so that can be expressed by looking at the computation graph for node U. Computation graph for node U is defined by this local neighborhood that defines the neighborhood that when messages are propagated along edges in that computation graph, produce embedding for on node U. And we can then essentially derive a formula that will tell us for any node in that neighborhood, we can see, well, what is the impact or the influence of V on node U. Now, a few subtle considerations here is that the size of those computation graphs often coincide with the number of layers what we have in graph neural networks, and so that has been the case in some architectures. In others, these local computation graphs are not necessarily limited to full K-hop neighborhoods of a particular node U. So that's why we refer to them as local subgraphs rather than K-hop neighborhoods, because practically they are not K-hop neighborhoods, as they can often be much larger than what we can computationally handle. You might be wondering on what is the value of subgraphs, and so the way the reason why we have relied on using subgraphs to transfer information from one task to the other to enable few-shot learning is the following. We often think of two sources of predictive power in graphs. The first source that many algorithms rely on is that of label propagation, where the algorithms often make an assumption that nodes with the same label are nearby in the graph. And because of that, we can effectively propagate labels along edges of the graph to predict labels for the unlabeled nodes. The other notion that is complementary to label propagation is structural similarity, where nodes with the same label can have similar network shapes or similar topology in their respective local neighborhoods, but those nodes can actually be very far away from each other in graph. So our reasoning was that when labels are scarce, then label propagation is not sufficient. Where we have only a handful of nodes labeled, it is challenging to effectively propagate labels to the entire graph. And so it would also be very challenging to learn these signature functions that describe every task at the entire graph level. And we cannot describe them at the individual node level. So subgraph level structure is then the appropriate scale at which we can consider and describe those tasks, and that allow us to much better leverage structural similarity, because the signal we are getting from a label propagation or the neural message passing scheme is very limited. And so next, I will describe one particular application of GMeta approach in biomedical discovery, and that will relate to the problem of drug development and in particular finding cures for a novel emerging disease. So the motivation for that work was observation that is not foreign to probably anyone here on the call, but when we were faced with pandemics last year, it was clear that there was a strong need to develop safe and effective therapeutics at unprecedented speed. So very quickly, and doing that in a way that would lend itself to useful therapeutics in a matter of months rather than years. And so this was challenging because traditional iterative development and experimental clinical testing of new drugs was simply not feasible. The discovery and development of a new drug from scratch takes on average 11 to 16 years and costs between $2 and $3 billion. So in the times of a health crisis, it's important to be able to really compress years of work into months or even weeks. And so the question is, can we do that? How we can do that? And so what I will outline is a project that we did as part of the COVID-19 task force, where we were able to compress this tremendous amount of work through the use of AI and new data sources into months and even weeks in certain cases. And the work that we did took place between March 23rd and June 15 last year. That's where majority of the work was done. And then during that time, we were able to collect diverse data sets, use GMeta algorithm to identify promising therapeutic opportunities for COVID-19, and then take most confidence and trustworthy algorithmic predictions and validate them in the lab and in human cells, which was done by our collaborators. And we published this approach recently in PNAS, and I will outline the work and where GMeta and how GMeta was used. So fundamentally, the question for this project was what drug treats what disease. So we wanted to have a predictor that will predict what diseases a particular chemical compound might treat. And so that's very challenging, because in order to understand drug-disease relationship, in particular drug-disease treatment relationships, there are a variety of different things we need to consider, because the way drugs work is not just by binding to a particular target protein, but then those effects propagate through the underlying biological network. So this is fundamentally then a network biology type of question. where we have this underlying biological network where I will describe it, what it was, and on top of it, we want to predict links or edges between diseases and drugs, where the kind of disease that we will be particularly focused on here will be COVID, we will be interested in is COVID-19. Okay, so how can we represent and how will we represent computationally COVID-19? So what we did was the following. We considered three sources of information. So the first one was the human-human protein interaction network. This is a network where nodes are proteins, edges between proteins indicate physical contacts between proteins in cells. The second type was information were viral human protein-protein interactions. These are interactions between the proteins in the virus and human proteins. So essentially it tells us what this information tells us, what are the human proteins that are attacked by the virus. And the third piece of information are interactions between drugs and proteins that tell us what are the proteins that a particular drug, when a patient takes it, affects. And so the way this can be thought of is then we have this underlying human interactor or human protein interaction network, and we on top of it, on top of that network, we overlay these small disease and these small modules or subgraphs that are defined based on whether what proteins are targeted by the drug, that would constitute the drug module, what proteins are attacked by the virus, that would constitute viral disease module. And in particular, that viral disease module is then our COVID representation of COVID. The COVID module was a subgraph of 332 human proteins that were shown experimentally as being proteins in humans that the virus attacks. So given information given this COVID-19 disease module and modules for many other diseases that we constructed, we formulated the setup in the data set in the following manner. So we constructed this large graph, which we call COVID-19 repurposing knowledge graph. It has this the underlying base, which is the human protein interaction graph. On top of it, we overlay these drug modules, and we have a separate drug module for each and every drug approved in the US. It is defined as a subgraph induced on the PPI network based on what proteins are targeted by the drug. We have a large number of disease modules. In particular, we had tens of thousands of modules representing common diseases, and one of those modules was also COVID-19. And for those common diseases, such as different kinds of cancers, cardiovascular diseases, and other infectious diseases, we know what their drug treatments are. So the task then was, given those common diseases, their disease modules, and treatments for them, how can we use that information to identify candidate treatments for COVID-19? And so this is a zero-shot learning problem. Let me explain that. Based on the terminology I introduced earlier, zero-shot learning refers to the setting where we have zero labeled examples for training. This is what we had here, because for COVID-19, we didn't have any successful example of treatment that we could use for prediction. We had, however, successful examples of treatments that are already developed for common diseases. So the way GMeta was used for this particular application was that in the meta training phase, we defined a large number of training tasks. Each training task was concerned with identifying or training a predictor that can distinguish between drugs that are successful for one disease versus drugs that are not successful, not treating that disease. So training task 1 would be concerned with identifying treatments for disease 1. Training task 2, it would be concerned with identifying treatments for disease 2, and so on and so on, for every common disease. And doing that in the meta training phase allowed us that in the meta testing phase, we were able to prioritize drugs for COVID-19, even though no examples of successful treatment, so no examples of drugs that would treat COVID-19 were available. So the first the first test for us was to use GMeta to produce predictions and then look at what are drugs that GMeta identified as most promising drugs for COVID-19. And at that time, we looked at we took those drugs and compared them against those that were in clinical trials for COVID-19. And so this is the results of that analysis are shown on the left part of the slide, where we are showing the ROC plot, and in particular in this grayish shade are shown true positive versus false positive rate curves for GMeta, compared to alternative approaches, which in our case were based on random walks, and then different variants of random walks, D1 to D5, and different notions of network proximity based on shortest path calculation, which were predictors P1 to P3. What we found is that GMeta was able to much more accurately than those alternative approaches identify what are most promising drugs based on what biomedical community thought were promising drugs and were pursuing clinical trials for those drugs. However, just the mere fact that the drug is in clinical trial does not mean that that drug will actually treat the disease. So to go one step further, we engaged in experimental screening of top predictors made by the algorithm. And in this particular case, our collaborators from the National Emerging Infectious Disease Laboratories screened 918 compounds, drugs that were predicted with the strongest confidence by the algorithm. They screened those drugs for their efficacy against SARS-CoV-2 virus, first in African monkey cells and then in human cells. The results of that experiment were that by screening top-ranked drugs in human cells, we obtained a 62% success rate. What does that mean? That 62% of the drugs that were predicted by the algorithm and tested in the lab were indeed shown to induce viral growth. In contrast, with just 0.8% hit rate that would be obtained if one would use non-guided screening, meaning just exhaustively test out all the drugs in a non-prioritized manner guided by the algorithm. And so this is an order of magnitude higher hit rate among the top drugs than what would be possible to obtain by these alternative strategies. And so we also wanted to understand what is there anything common to those drugs that were successfully predicted and validated in the lab that they indeed inhibit viral growth? Are there any common mechanisms to those drugs in the way how they exert their therapeutic action? And so to be able to do that, we relied on an algorithm we recently developed. It's called GNN Explainer, that is a post hoc tool that can take a particular prediction made by a GNN model, in this case GMeta, and the model itself, and it can identify a small subgraph that is most influential for the prediction made by the model. And so using GNN Explainer on our data set and allowed us to ask the kind of questions such as, will a form of a drug, a statin, treat hyperlipidemia? What is the disease treatment mechanism? If that's a question that domain users ask us or ask the model, then the GNN Explainer would return a small subgraph that consist of human proteins, biological processes, drugs, and disease in question that represent a biomedical knowledge that is most important and that was used by the model when making that prediction. And so we generated those explanations for COVID-19 predictions and examined them and tried to identify what is common to those drugs that were successfully predicted and validated. And so the results of that was that we found that 76 out of 77 drugs that were predicted and successfully validated and shown to reduce viral infection were those drugs that do not bind directly and proteins that are attacked by the virus. So instead, those drugs, which we now refer to as network drugs, they exert their therapeutic action by targeting human proteins that are not attacked directly by the virus, but those human proteins then in turn interact with other proteins that are eventually proteins that are attacked by the virus. So the implication of that is that this group of drugs clearly really rely on network-based actions and could not be identified by docking-based strategies, which is a typical approach for drug repurposing in COVID, in broadly speaking. And so this indicates really the important use of network-based algorithms for drug discovery. Okay, so that concludes the first part of the talk. And so in the remaining few minutes that I have, I will also go through the key points of the second part, which is focusing more on infusing knowledge into biomedical AI algorithms in the sense of additional side information and then how that can be leveraged to, in our case, solve patient-related problems, in particular diagnosing diseases. So the problem formulation for this particular part, for this part of the talk is as follows. So here we will also learn about and leverage subgraphs, but we will use them in a completely different manner. So if you remember in the first part, GMeta, we used subgraphs not because inherently we were interested in subgraph modeling, but we used subgraphs as a mechanism to extract, to describe structure of different learning tasks and then transfer information from one learning task to the other. In this part of the talk, we will use subgraphs in a different way. We will use subgraphs to encode additional information or knowledge into our representations. So the goal here will be that when we will operate with the assumption that our input graph is a graph that can be annotated, and it also contains a number of subgraphs that are overlaid on the graph. So imagine this as input being this graph G, which is this underlying graph where nodes are shown as white dots, and on top of that graph we have different subgraphs. S1, S2, S3, S4, S5 are examples of those subgraphs. And then the goal really is that we want to predict and label these subgraphs rather than individual nodes. And so the way to do that in the framing of subgraph representation learning, our goal is to learn subgraph embeddings such that the likelihood of preserving subgraph topology is maximized in the embedding space, meaning that if we have two subgraphs S sub I and S sub J that have similar topology, they should be embedded close together in the embedding space, and we will then be predicting labels for those subgraphs. Those subgraphs can be, for example, patients. They can be, for example, different clinical settings that we want to label. So one note on this problem formulation. So this problem formulation is.
Hey,
yes.
Can I also ask one more clarifying question? One is, what happens if the graph is not, a part of the graph is not connected to another part?
That's a great point. So what it can happen is we assume that the base graph is connected, but subgraphs do not need to be connected. Like if you see here S1 actually it has two components. This is this green subgraph. It has two components. These components on 4 nodes here and another component on 4 nodes here. And the same for S4. And that is what happens very, very often in real practice. And so the method that I will describe takes care of that. It has a specific component that allows us to operate with these subgraphs or signals on graphs that can be disconnected.
Great. And another one was that here and I don't know whether this is correct or not, you assume that the interactions between the nodes are symmetric, right? And that's why there is no direction. But I can assume that, if especially for drug discovery and other things, the interaction can be one way, right? This protein might have some influence on the other one, but not the other way around.
Yes, that is correct. Directionality is often important in drug discovery problems. So in the second part of the talk, I will talk about patient diagnosis, and the data there most often comes in the form of associations between patient phenotype and genes mutated in patient or drugs that the patient takes, and those are symmetric relationships. In the first part, when we're talking about drug discovery, many edges there or relationships are by design directed. Often when we develop models for those data, we consider those relations undirected. Why? This is not solely because the lack of models for directed data, and there is a gap in graph machine learning models, in particular graph neural networks that could operate with directed data sets, because it's much harder to propagate neural message passing if you are following directions of edges. In the drug discovery space, particular challenge is also the lack of reliable experimental data. So biotechnological platforms that are generated these readouts, measuring interactions between proteins and measuring their physical contacts. Today, on a large scale manner, those platforms cannot detect directionality of interaction. They can measure that two proteins are in contact at a certain point in time, but they cannot resolve that contact and identify which of the two proteins has initiated that contact so that they would be able to resolve directionality. And so that's the second reason why most of existing work, including the work that I described in the first part, ignored directionality between proteins.
Marinka, I have another question related to that. It's relevant for the first part of your talk and also second one. You assume the structure of the graph is given to you, but in many of the biological problems, creating the graph is an art of itself, because the graph is not something that God gave us, right? We are using some metric and we calculate connection between the different nodes. So do you do any experimentation when you do learning? Also, what is the structure of the graph?
Yeah, so that's a wonderful question. I'm not covering this topic in today's talk. It's very important and complementary to what I've been covering so far. So yes, you're absolutely correct. Both the topics that I mentioned today assume that we have a graph, and given that graph, the goal is to predict labels of individual nodes or links between nodes or labels of subgraphs in the case that I'm describing now. This is different from the problem of graph inference or graph construction. The problem of graph inference or graph construction until very recently could not be tackled very well with graph neural networks. But recently there is an emerging area of research known as latent graph learning that uses ideas from manifold learning and neural message passing to optimize for graph structure and construct a graph such that it will be most useful for downstream prediction tasks, or that it will represent a consensus over different kinds of measurements you might have. The part where that has been studied relatively well in the recent years was graph construction in the sense of small graphs representing molecules, but those methods are optimized for inferring very small graphs, because molecules as represented as graphs of atoms are often rather small. The work there has not been in this area of research has not been extended successfully to inferring very large graphs. So when we are concerned with this problem of large graph inference, for example, in single cell genomics, predominant approach is still to construct k nearest neighbor graphs based on some predefined metric of a similarity between gene vector profiles, and then once that graph is obtained, use it for downstream learning of embeddings and downstream prediction. Okay.
Thank you.
Let me try to define this problem in the remaining few minutes. So we were talking about the problem of learning subgraph embeddings with the goal of eventually predicting different properties for individual subgraphs. And so the problem formulation here is that we want to have a model that will take as input subgraphs, and it will embed subgraphs and represent them as points in the embedding space in such a way that two subgraphs that have similar topology, and I will define that next, what that means, should be embedded close together in the embedding space. So this formulation enables us to produce subgraph embeddings rather than embeddings of individual nodes. And in particular, it enables us to solve tasks when we need to predict properties of individual subgraphs. This is different from canonical tasks on graphs. Canonical tasks on graphs in machine learning are concerned primarily with predicting properties of individual nodes, of node pairs, or predicting properties of entire graphs, such as molecules. So why are subgraphs so challenging, and why is embedding subgraphs more than just taking an average embedding of all nodes that construct a subgraphs? Why is that? Why do we need to go beyond that? Because subgraphs are often that we want to predict labels for are such that require us to make predictions over structures of varying sizes. So those subgraphs do not necessarily correspond exactly to k-hop neighborhoods of individual nodes. They can be of different sizes and connectivities. When we look at subgraphs, they have rich connectivity patterns, and what I mean by that is that it's important to look at what is the internal structure of the subgraph, as well as how subgraph externally interacts with the rest of the graph that it is located in. And so the question then is, how can we inject that information into our message passing architecture that generates embeddings? And third, subgraphs can be localized in the sense that a particular subgraph would reside in one region of the graph and would form a connected component, or they can be very distributed across multiple local neighborhoods. And then how we can bring those local neighborhoods together, it's a question. So to address those challenges, we developed an approach that is called subgraph neural networks. The subgraph neural networks method, or sub-GNN method, and I won't be able to cover the details, but so I will just define it here, operates in the following manner. It takes as input a set of subgraphs, S1 up to Sn, and it specifies a neural message passing architectures that generates this S-dimensional subgraph representation or embedding for each and every subgraph. And then those representations can be readily used to learn subgraph classifiers and predict labels for subgraphs. Importantly, SubGNN produces those subgraph embeddings by taking into account 3 aspects of subgraph topology. And so by what the 3 aspects that subgraph neural networks consider are neighborhood, structure, and position. So what I mean by that is neighborhood refers to the information about the connectivity in the sense of the number of neighbors and their connectivity patterns for each and every node that constitutes a subgraph. Structure refers to the over or underrepresentation of certain network shapes in the subgraph. And position refers to the question of whether the subgraph is located close to the periphery of the graph or whether it's located in the core part of the graph. And each of these 3 aspects of subgraph topology can be studied from the standpoint of internal structure, internal neighborhood, internal position of the subgraph versus how it relates to the rest of the graph, which is the structure and neighborhood of its border. And SubGNN can produce those subgraph embeddings by disentangling these distinct aspects of subgraph topology. And to do that, it specifies a hierarchical neural message passing scheme that has 2 parts that essentially allow us to propagate neural messages along edges that participate in a given graph, and then aggregate that information that we get from each and every node that is part of the graph to the level of entire subgraph. I will not have time to go into details, but we described the SubGNN in detail in a paper that we published at NeurIPS last year. Perhaps I will conclude just with one example of how SubGNN can be used for disease diagnosis. And so the motivation for that application was that when the doctors are tasked with diagnosing a patient, they are making that diagnosis by not thinking of a patient as an individual node or an individual point, but really as a richer representation. For they would typically have information available about medical history, about symptoms of that patient, and that would need to be taken into account collectively when making predictions. And so that means that we want to model diseases not as individual nodes in the graph, but as collection of phenotypes or symptoms and diagnose patients based on that. And so effectively, that means that in the diagnosis task, we would have the following task that we would need to solve. We have this underlying knowledge graph, which in this case is human phenotype ontology, which is an ontology that has been manually curated that captures relationships between human phenotypes. And every patient would then be represented by a subgraph on top of that HPO network, where a subgraph is induced by the phenotypes or symptoms that the patient has. And given then a large number of those subgraphs in patients, we can use SubGNN to predict or classify subgraphs, which effectively mean that we are classifying and predicting diseases that patients have. And so that is an example use case of SubGNN that we're describing in the paper, in the NeurIPS paper, as well as many others that I won't have the time to cover. So I will stop here. Thank you very much for your attention. I'm very happy to take any questions if there is time. Otherwise, I'd be glad to point you to all the codes and data sets that we have available on our lab website. And I can also take questions now. Thank you.
Okay, thank you very much for this inspiring talk. So we have asked many questions during the talk, but if there are other questions, people can ask now. Go ahead, James.
Thanks for the talk, really nice. I'm just curious about the sub-GNN setup. Is there anything unique or interesting about the loss functions, the math of the loss functions, or is it pretty standard, or just curious?
Yeah, so that's a great question. The interesting part of the loss function is that we are using contrastive learning for the loss function. So intuitively, the idea is that for each of those three aspects of topology that I mentioned, we define an augmentation process that allow us to distinguish between, for example, the structure as we see it in our graph, subgraph that we want to embed, versus the structure in random patches, which are random subgraphs that we sample from this underlying base graph that we have. And so the goal is then to produce an embedding that is indicative of the structure of the subgraph, and that embedding is considered to be good if it can discriminate between the actual structure of the subgraph and the structure of random patches. And kind of the same idea is then also used for capturing position of the subgraph and its neighborhood.
And each one of those ideas is one term or something like that in the loss function. Is that the idea?
Yeah, so instead of having a loss function with multiple terms that often ends up being quite difficult to optimize because it's hard to select the weights of each of those terms, what we're using instead is three different channels in the neural architecture. So we don't have a single neural message passing channel or a single neural message passing algorithm, but we have three of those. One for each aspect of topology, a separate channel for neighborhood, a separate channel for structure, and a separate channel for position that allow us then to use different augmentation strategy for each of those three aspects, and also allow us to be able to effectively combine these three aspects, have these three channels without the need to do more hyperparameter selection in the sense of the weight of each of the term in the loss function.
Yeah, thanks. It's interesting.
Okay, so I think we are running out of time. It has been a very, very interesting talk, and as Marinka said, there are, I think, all sorts of information available on her webpage. So let's thank her again, and we hope that we'll see you soon in person.
Absolutely, thank you for the invitation.
Have a very good day.